Thank you very much. It is my privilege to talk to you about the Collaborative African Genomics Network, which we call CAfGEN, and the 5 institutions that are collaborating in this regard are listed here. And many of the PIs are in the room, and they will be available to answer all the difficult questions after my presentation. So I will just take maybe about 2 or 3 minutes each to address each one of those bulleted points. I think everybody in the room is aware that HIV continues to be a scourge. There is no country in the world that is spared. But critically for us, Africa, especially sub-Saharan Africa, is the worst affected region in the world. And although HIV has peaked in many countries, as represented here by Botswana, and seems to be declining in some other countries, as exemplified by Uganda, it dipped and then it's beginning to rise again, and it seems HIV will be around for a long time. The natural history of HIV is extremely variable, but it's characterized by 3 phases. An acute phase where following infection where there is extremely high viremia and where the CD4 cell count is still high, which lasts in an average adult about 12 weeks. And during which time the person may experience a flu-like illness, which then settles down into a latent, clinically latent phase, which may last several years. And although things appear to be normal on the surface and the person may be clinically stable, the CD4 cell count is during this time beginning to decline until it reaches a level where they begin to develop constitutional symptoms and eventually serious opportunistic illnesses that constitute what we call AIDS, which without treatment culminates in death. In children, these phases are really extremely variable and very much depend on the age of the child. In the majority of children who get infection at birth, symptoms actually develop from around 3 months following infection, and without treatment, up to half of the children may be dead already by 1 year, and only 15% survive 4 years and above. But in a small proportion of children who we call long-term non-progressors, the clinical history is more like that in adults. And these children are able to contain the virus for a long time, and they only begin to develop symptoms after about 10 years or so. Now, regarding TB, the prevalence of TB was declining until about 30 years ago when the HIV epidemic started, and then it too began to increase. Sub-Saharan Africa in general, and Uganda and Botswana in particular, are very severely affected, as you can see from these notification rates here. TB, like HIV, has a clinical latency as well, following infection, and the period of clinical latency is extremely variable, depending on the nutritional status of the person and the immunity of the person. For HIV-negative people with normal immunity, the lifetime risk of progressing from latent to active TB is 5 to 10%. But in HIV-infected people, the annual risk is 5 to 10%. HIV has a huge impact on clinical latency and makes it extremely rapid, makes it extremely short. Overall, among adults with HIV, about 60 to 80% of them are co-infected with TB. And when it comes to genetics, well, this has the potential to transform our understanding of these diseases, TB and HIV. But paradoxically, most such studies have been conducted in non-African adult populations. Studies among HIV, children with HIV have been conspicuously absent. HIV-infected children differ, as you have seen, from their adult counterparts and have ultimately more to contribute and more to gain from therapeutic advances. So the mission and vision of CAfGEN is shown here on this slide. The vision is simply a future where African scientists are using genomics and similar techniques to ameliorate the health problems on their continent. And our mission is to create, as part of the H3 Africa Consortium, a collaborative, multi-disciplinary, multi-institutional, inter- and intra-country network of African scientists, clinicians, and researchers to use genomics approaches to study gene-pathogen interactions for HIV and HIV/AIDS, its co-morbidities and other diseases among diverse pediatric African populations. The partnership with CAfGEN is based on multi-layered existing relationships between the participating institutions, which have been going on for a very long time, and many of which are governed by memoranda of agreement. The specific aims are 5. First, we aim to recruit well-phenotyped pediatric HIV and HIV/TB infected patients and create a DNA and RNA biorepository from blood and sputum samples that will be linked to a central clinical database. The second aim is to evaluate the roles of established and novel HIV disease progression alleles in children by sequencing and allelotyping candidate genes and by using whole exome sequencing in case-controlled genetic studies of long-term non-progressive status. The third is to use integrated studies of clinical outcomes, DNA and paired RNA analysis in HIV/TB co-infected children to identify genes that contribute to the progression to active TB. The third is to enhance undergraduate and graduate faculty education in genetics genomics and to provide opportunities for long and short-term training of scientists and technicians from African universities. And the fifth, but not least, is to establish genetic and genomic technologies and supporting laboratory and physical infrastructure so that these can be used in a large-scale analysis of common diseases in Africa. The strategy is illustrated in this schematic. Essentially, it will be to use 3 core projects simultaneously to address all the 5 aims that I've just listed. The tan-colored steps shown here indicate areas where major infrastructural developments are expected to take place. So, in the core project 1 on HIV disease progression, we will recruit a cohort of 500 using electronic medical record, which is common to the recruiting centers in Botswana and Uganda, and with which we have had a long-term experience. This, the retrospective HIV cohort will be defined as indicated here. The long-term progressors will be children who will be asymptomatic for HIV for more than 10 years from infection, which they will have acquired at birth without treatment. And rapid progressors will be children who will have had at least 2 CD4 cell counts with values less than 15% without ART. These cohorts will include discovery and validation patients as well. And in the prospective study for HIV, we will recruit 2,000 HIV-infected children and then follow these over time up to 18 months to 2 years for progression to active TB. Over the period of about 18 months, we expect approximately 150 of these to progress to TB. And then we will also recruit an additional cohort of HIV-infected children with active TB at first encounter for validation studies. And this is explained differently using this schematic here. This is the 2,000 patients that we hope to follow for 18 months. At baseline, we'll collect blood for RNA, we collect clinical data, and also blood for DNA. And when they develop TB, we'll also collect blood for RNA, clinical data, and induce sputum if they are not able to produce the sputum themselves, and use that for microscopy and culture and gene expert to confirm the diagnosis of TB. In the approach in the retrospective cohort is to identify host genetic variants within the coding regions of candidate genes and throughout the genome that influence disease progression, by first assessing the genes that have already been associated with the HIV disease progression in other populations, and then using whole genome, whole exome sequencing, identify new host genes affecting pediatric HIV disease progression by looking for sequence variants enriched in long-term non-progressors. Then promising variants will then be replicated in the remaining 300 long-term non-progressors and 300 rapid progressors. As shown in this schematic, what is important to indicate here is that these activities are going to be done at the University of Botswana, at Makerere University, and at Vega College of Medicine, mainly by the trainees. And so the specimens really will be sent to Beta while the trainees are there for training, and the trainees will continue to work with these specimens as they continue to be trained in this particular area. The aim of the prospective study is to identify genes that show differential expression with progression to active TB disease in HIV co-infected children. We will use microarray genotyping combined with RNA sequencing to look for SNPs that are linked to changes in messenger RNA expression during the progression to active TB in these children. Strong candidate genes must show both a change, an increase or decrease in expression during this transition to active disease, and the level of expression of gene must be influenced by the SNP genotype. And this will be, is also again shown in this schematic, is summarized in this schematic. The data from data and sample flow is shown in this schematic. We will use a web-based LIMS, which we have just about finalized, which will feed into the CAfGEN database servers, which will feed into the H3 Africa bioinformatics, and eventually into public databases. We have already created a mirror image, mirror EMR for this purpose, and which has been tested in Uganda and is currently being tested in Botswana. The training network is shown in this schematic. Essentially, the identified graduate PhD students will undergo long-term training at Baylor College of Medicine for about 2 years. They will train in next-gen sequencing, genotyping, bioinformatics, statistical genomics, and so on. The training will include both coursework, symposia, and they will also have Baylor College of Medicine certification, which will count towards their PhD requirements. They will also, on return, be involved in LC short-term courses and training in local communities. So in effect, the training network is going to be complex, and the strings that connect these different boxes here indicate the growth of the students who will be the most important part of this training network. So the progress to date is that we were funded by NIH in December 2013, so just about 5 months ago. We have obtained 4 IRB approvals. There is one that is pending at the University of Botswana. We have essentially completed staff hiring and training. We have finalized standard operating procedures, which we have made in line with those of the H3 Africa guidelines. We have identified 2 students. One is already gone to Baylor, and one will be joining next month to start their training in genomics. We have already started the process of purchasing equipment, and I think some will be delivered in Uganda very soon. It hasn't been delivered already, and as well as in Makerere, and pieces which have been ordered indicated here. We have partnered with Oric Harvard Law School to conduct a desk review of relevant LC literature in order to compile best practices, which we are going to feed in this training network. Next steps to conduct dry runs as soon as we get the approval from the University of Botswana. And then we have already identified from our EMR all the patients that we need to, since we see them very frequently, as soon as we obtain IRB approval, recruitment should be pretty rapid. And we expect to recruit the 500 long-term non-progressors and 500 rapid progressors within 5 months within obtaining IRB approval. The establishment of a genomics training program at UB and Makerere is is under the works, and activation of a community advisory board so that we can have wider community engagement is also a planned step. We have identified membership of this from the, which is going to consist of local chiefs. They are the most important people at community level, from the House of Chiefs. Not the House of Parliament, people circulate every 5 years, but the House of Chiefs is permanent. We'll also have people representing major religious groups, major HIV NGOs, and local IRPs. We will be monitoring the trainees as they go through this process very carefully, and we have engaged in steps to source extra funding so that we can carry out community and media workshops to prepare them for this type of research, because it's relatively new. And we also are in the process of working with others to publish a comic book to inform the general public on matters related to genomics research. So in conclusion, I just want to thank all the people who are listed here on this slide, my co-principal investigators who are in this room, as well as co-investigators, and special thanks to Team H3 Africa for organizing this meeting, the H3 BioNet, the Wellcome Trust, and the NIH. And especially our program officer who is attending this meeting for the first time, Sudash Universal.